GSE 49110 is a dataset that examines the effect of knocking down the ER-RALFA gene using 3 different siRNAs. Including the control, there are 4 groups, each with 2 biological replicates. On the Subio website, go to the Support menu, select How to use Subio, and search for RNA-Seq. Open the article titled, Import RNA-Seq Gene Count Data from GEO, and copy the URL. Replace the GEO accession number in the URL with GSE 49110 and open the page. From that page, download the NCBI generated raw counts data file. Go back to the GSE 49110 page, scroll down, and download the soft formatted family file. Next, open the NCBI FTP site. Navigate to Gene, Data, Gene_info, Mammalia and download the Human Gene Info file. Go back to the Data folder and download the Gene2Go file. This file is large and may take some time. Finally, open the UCSC Genome browser and go to the Table Browser page. Make sure the assembly is set to GRCH38 and select the NCBI RefSeq track. Specify the output file name and click Get Output. You should now have downloaded 5 files. Extract all files except Family, Soft, GZ. Open Excel and drag and drop the Human Gene Info file into it. Select the entire table and sort by the type_of_gene column. Search for protein coding in the type_of_gene column. Delete all rows that appear before the protein coding entries. Then search for pseudo in the same column. Delete all rows from pseudo onward and save the file. This leaves only protein coding genes. Open the Data Manager tab in Subio Platform and click the New Platform button in the toolbar. Drag and drop the edited gene info file into the window. Enter a name, and proceed. Double-click the Gene ID column and set it as the measurement ID for the platform. Your platform is now created. Since gene IDs are not intuitive, select the gene symbol column for display. It will be highlighted in the title list. Click the Edit Platform button to open the editing window. Click Look Up, and drag and drop the Gene2Go file into the dialog. In both tables, select the Gene ID column as the matching key. Select the column to import. Here, choose the GoTerm column. Make sure to enable Concatenate All Values. A GoTerm column has been added, with multiple values separated by. Use the lookup tool again, this time with the RNA-Seq genes HG38 file. This time, use gene symbol as the matching key in both tables. Import the following columns: CROM, strand, TX start, and TXEND. The TX start and TXEND columns are appended at the end. Copy all values from TX start and TXEND, then paste them into CROM start and CROM end. Scroll down to confirm that all values were copied correctly. Delete the TX start and TXEND columns, as they are no longer needed. You can reuse this platform in the future, so this setup is only needed once. Click the Import Samples button and drag and drop the raw counts file. Use the Multiple Samples in One File option to extract individual samples. 8 samples are listed. Proceed to complete the sample import. You may notice that the number of rows is reduced compared to the original file. At this stage, non-coding RNAs have been filtered out. Since GeoSample IDs are not very informative, import metadata from the soft family file. In the editing window, select any cell in the file name column, split the values at the dot, and remove the .txt part. Click Lookup, and drag and drop the soft family GZ file. Use the GeoSample ID columns in both tables as the matching key. Select all columns to import. Delete the temporary sample ID column used for matching, then finish editing. Since there is now too much information, uncheck columns you don't need. Then check only the columns you want to display.